Hi everybody. In this video I wanted to go through some of the key things that we talked about during the first few weeks of the semester in this module. So I gave you an overview that you can access online with kind of the topics we're going to talk about at the start of the module. But effectively the first section we went was really focusing on the biopharmaceutical industry, how things were produced, some key aspects, and then the development in recent years of biosimilars. So we looked at the comparison in size between these products, looking at the size of the antibodies. These protein drugs are very large compared to sort of chemical therapeutics that are available currently. We talked about how they're a lot more complex. We talked about a lot more kind of regulatory checks and so on, just with regards to even their structure. And that's a lot because they came from a living entity, this biopharmaceutical and that living aspect. So we use recombinant DNA technology to build these products. So we went through the general plan of how we get genes into vectors or plasmids, which then can be transformed or transfected into cells known as expression systems. And we talked about sort of E. coli being used here, say for in the production of insulin. And then we talked about other proteins which require the idea of large bioreactors for production. So we focused a little bit on insulin as a nice example, something you were familiar with and something you've talked about later on in the module as well. But just the idea that bacteria could be used here and how this was, I suppose, and a good example to work through because it was the first kind of therapeutic produced in this way in 1982 by Genentech, this humulin that was produced. And then we scaled up a little bit to producing tissue plasminogen activator in Chinese hamster ovary cells, which happened 5 years later at the time. And this idea of getting, instead of going into E. coli, you go into an expression system known as Chinese hamster ovary cells, these CHO, and these are the most predominant manufacturing system, I suppose, that are used for a lot of therapeutics. They're able to fold proteins in a particular way that works for human patients, and they're also able to add sugars and glycosylate them to the extent that we need for the patient's, I suppose, for this drug to work with the patient. So then we looked a little bit about biosimilars in recent times and how they're produced and what they are. We watched a little animation that I think we all found very helpful, so I'd recommend going back looking at that. And then we talked about the where they fit into the system with regards to different comparisons, I suppose, with their approvals. We talked about a thing called reverse engineering and how reverse engineering is what can be done before patents expire. So learn a lot about biosimilars and develop your own. And then we talked about all those different steps that are involved in that production of this biosimilar entity. So we saw it's something actually that is going to overtake even the production of the biologics here, these biosimilars production over time, that's predicted to make that change. Then I guess I went back and I gave you a list of different things that we could think about, how things were made, how do we get them, why are we studying them. Just to even, if you go back through the notes, I often gave you a few exercises to kind of reflect on, like even in biosimilars here, I asked you to draw a flow chart of this process so you can reflect on the notes and work that you did during the term. With regards to expression systems, we rated them based on different criteria and sorted them in that order, and we discussed why we would pick one over the other. We looked at the process in a kind of a flowchart fashion, probably coming from the diagram before, the same process, but just in a kind of a flowchart, so we understood each aspect. And once we knew kind of the difference, I suppose, between biopharmaceutical and pharmaceutical expression systems, folding, glycosylation, and so on, we then moved into a family of proteins and biologics that we were going to look at. But first, I wanted to just give you a real overview of what they entailed. So we were going to talk about cytokines in general and how they work through receptors. They cause internal signaling and then they have a biological effect. So I wanted to do this at this stage because I knew in the second half of the semester you'd be doing a little bit more about signaling. So I thought if we could focus a little bit on that now and get the principles of it, then you'd be able to apply those as you kept studying throughout the module. So in general, we learned they were chemical communication messengers between cells. You've told me a lot about that already from your immunology work. We saw how they are essentially molecules that can be secreted by one cell, travel to another cell, and dock onto a receptor and cause an effect to occur in that target cell. So we learned about the phrases autocrine, paracrine, and endocrine, and how these can actually travel to work. we learned about how the cytokine or the signaling molecule binds to a receptor, activates relay proteins that send a signal that ultimately gets into the nucleus and can activate specific genes in order to have a biological effect, so that we see the effect of that particular molecule in the target cell then. So we talked about signals being amplified, how we can bind to a receptor, but then we get a huge amplification of the signal in the response that we want to see. So it was really just getting that principle of something like, say, a hormone, working in an endocrine fashion, traveling, binding to a target receptor, passaging the signal through the cell until we get a response. So the reception, the transduction, and the response pathway. And once we kind of had those principles, we were able to focus on a family of cytokines. So this term we looked at interferons, and we looked at their general 4 activities, what their roles are. We looked at the idea of the interferons. Once a cell got infected by a virus, we would produce that interferon protein, which got secreted. And here, this was the important point here, that we would have a receptor, a signal transduction pathway, produce different or turn on specific genes, produce certain proteins, and then have a biological effect. So this signaling pathway was what we were going to learn a bit more about this part of the diagram. So we talked about 3 different classes. We went through each of those, and we began to look at, again, this idea of what signaling could happen. So the cell that's secreted, the receptor, the transduction, transcription factor now can get into the nucleus once it's activated and switch on specific genes. Not every gene, specific genes that would then be transcribed and translated into a protein that would have a biological effect on this target cell. So when we looked at this diagram, it was very busy, but the idea was that you have various ligands that bind to receptors, they have signaling and transcription factors. So you begin to see the kind of pattern that's emerging here, where we were going to lock on here to cytokines. These cytokines that work on cytokine receptors, they use particular signaling pathways, and they have transcription factors. So JAK kinase and STAT proteins were the signaling pathway and transcription factor we were going to focus on. So we looked at some basic diagrams of the JAK-STAT pathway, and here we talked through the process bit by bit using the text that associated beside it there. And then we did a little flowchart that kind of worked us through that diagram a bit more. So now we knew how the signal was processed into the nucleus from that interferon. We learned that the transcription factors, once they get in, bind to specific sequences known as response elements. And these response elements are the landing sites for specific transcription factors in specific genes. So those specific genes get switched on in specific circumstances, such as in response to an emergency or along those lines. So TTCCX, any base there, GGAA, this sequence is almost a landing site for, say, the STAT molecule, the activated STAT, and it will be located in the promoters of specific genes. So we talked a little bit about those response elements and what they were. We had touched on it in second year, but we were reminding ourselves of what they were and how it would work here. Then I asked you about the biological effects of interferons, and I showed you here a collection from the 2019 class where they collated all of the different points that were in a handout that they could find. So I asked you to go back and see, when you went through the handout, could you modify or increase these, or even add a few lines around each one? These are bullet points, but could you describe or understand a few of these, as in what will be the biological effect that you see? So interferon binds to receptor, JAK-STAT pathway turns on specific genes that make specific proteins. What do those proteins do? That was essentially what we're seeing here. When we had gone through the interferons, we kind of changed gear into a new section then, into our second handout, and it was on EPO and these hematopoietic growth factors. So EPO is another biologic that can be made. RH-EPO is recombinant human erythropoietin. But first we learned a little bit about how stem cells and how they get differentiated based on the cues that they receive. So we were going to look over here at this side and the formation of erythrocytes. And erythropoietin, the target we're going to look at, works on these CFUEs primarily in converting them to erythrocytes. So we had to learn a lot about stem cells and differentiation first. Our focus was EPO. We looked at how it's produced in adults and in the fetus. We saw how, this diagram in the top left is a lovely summary of what actually happens when we have low oxygen detected. We'll go into more detail in some of the other slides. But effectively, we saw all the different cells that are responsible, I suppose, for those production of the erythrocytes. And we saw CFUE was a cell that had the most amount of receptors on the outside to meet this EPO molecule that's produced or bind that it can bind to, and ultimately help those cells accelerate the production of erythrocytes. So this was a nice slide as well, because it just showed us a lot of these interleukin 3, GM-CSF, interleukin 6, interleukin 5. We saw that these were these cues that were being docked onto different cells, and it could guide them in particular ways. And even how the threshold, the amount of each one, can decide if they're going to go one way or another. A lot of our focus was around the idea of how hypoxia works, and how we have something called a heme protein that acts as an oxygen sensor that can be either oxygenated or deoxygenated. So this concept was just, how does EPO actually work in hypoxic environments? So we learned about the natural in the body, the natural causes, how we can have an oxygenated form and everything's okay, but in a hypoxic environment we can have a deoxygenated form that triggers cellular events, transcription factors going through the nuclear membrane, landing on response elements and turning on the EPO gene. And we kind of followed these arrows all the way around and summarized it in this flowchart. So if we start up on the left, we saw hypoxic conditions, and we went down through the exact way it works, learning a bit about how the HIF is involved in this production, this transcription factor. So it built a little bit more on our signaling and turning on specific genes in specific situations. And I think learning the different players that were involved gave us an idea about how we could reflect on proteins being degraded, or else proteins being maintained in their structure, and then being able to function and team up to look for these response elements to turn on particular genes. So we were just, I suppose, discussing that idea about why patients would get EPO and what the EPO would do and where it would work. But we had to learn a little bit about how it functioned. We saw here the idea of some of the therapeutics. We saw disulfide bridges, so it made us reflect on protein structure. And then we saw these little dots and these little trees, and it made us focus and talk about N-glycosylation on asparagines and our serines and arthrenines that can be O-linked with our glycosylation. So we can have N-linked and O-linked glycosylations. But one of the reasons we look at EPO in particular is because this is a molecule where if we get rid of the O-linked sugars, things work in vivo and work in vitro the same. It works really well. But if we get rid of this in vivo, the O-linked, everything's okay. But if you get rid of the N-linked on the asparagine residues and you remove the glycosylation, there is hepatic clearance. So your liver actually removes this in minutes compared to the normal 4 to 6 hours. So it's vital that we kind of reflect a little bit on glycosylation when we're making a biologic or a biotherapeutic, and not just the protein shape, but actually the tags that are on the outside of that protein. So that was one idea of one focus that we talked a little bit about, even reflecting a bit on biosimilars here. What would you change to try and make the therapeutic even better or improve it for a patient? So then we went on to our last topic in the third handout, which was to do with monoclonal antibodies. And we went through this diagram about how we can produce monoclonal antibodies, or how they were started to be produced, I suppose, initially. We talked about the problem of having mouse antibodies. If they were all mouse proteins, what would happen? That people would reject them, and how we'd much prefer to have chimeric or humanized antibodies that have, the gray indicates the human aspects here, and some elements from the cells that were originated from the mouse that could find the target protein. That way our antibodies wouldn't be rejected, or we wouldn't be raising antibodies against mouse antibodies. And then secondly, we would also be able to use these antibodies with human FC receptors on macrophages, neutrophils, so on. And we maintain the antigen binding capacity from the mouse, traits that were gained by those cells. So we looked at I guess, how to humanize more, other methods to do it, how to make them more human. And we talked a little bit about this idea of a human artificial chromosome, and we generate certain mice that will make humanized antibodies for us. Much more because we give them the human information to work with. Another example was this idea of phage display. We can make different combinations of variable domains. The genetics, we can get them expressed so that they appear on the outside of certain phage up here. And then we could find out what targets bind to what phage and where we get a hit. We basically repeat this process over and over, and then we can find the genetic combination that makes a really good variable domain, and we can fuse that onto a human FC region and ultimately try and make, or down here, it's pretty clear here, we have our variable domain protein folded that we know works. Well, we can try and fuse that onto the FC receptor, because we know that this combination, say in the middle, is the one that makes the shape of the antigen binding region that works really, really well and finds the antigen. So once we know that gene that's in here that makes the particular folded region on the outside, we know that's what we want stuck onto the end of our antibody. But the more human constant regions we can make, the better for the long-term success of the therapeutic. So we went through, I suppose, a few different ideas with regards to how that hybridoma concept worked and fusing things together. How do we select? We talked about that hat medium, how they were produced, and how we can learn a little bit from hybridoma cells, and ultimately making sure that these antibodies are less blue, as in less mouse, all mouse, and that we try and make them more humanized in that concept. So we can learn a lot from generating our antibodies a particular way. And I guess our main focus here was about the idea of these monoclonal, finding one target on one antigen. And then on the top, you can see here blue, and very little blue, but mainly gray, because that's more humanized. And one of the reasons was, well, our immune systems will obviously not raise a human anti-mouse antibody response, but secondly, our cells will be able to recognize the receptors on certain immune cells that are called FC receptors, will look for human FC regions. So if our new therapeutic finds the target here in the middle, well, our immune cells will be able to detect that very well because of that recognition that can take place. We then began to think a little bit about antibodies are great and they can label things for destruction, but they don't kill a target cell themselves. We need the immune system to be able to find them. And that came back to that FC receptor discussion. But we also thought, well, could we conjugate things onto the antibodies? Could we conjugate drugs or toxins or radioisotopes so that the antibody brings to the target cell a sort of a death signal that could try and kill the target cell? And we make it very specific so it lands on that area. So we began to think about these immunoconjugates and what could be stuck onto different antibodies. And we talked about the linkers that have to be involved and the different types of linkers that can be introduced in adding a therapeutic onto this biologic that's being generated. So we looked at the idea that some cancer cells express unique surface antigens on the outside, and then the antibodies are able to find those that we make. But if we could conjugate something on the outside, like a radionucleotide or a toxin, they might be able to affect the cancerous target cells and leave healthy cells alone. So we focused on this little block here an awful lot. We talked about the energy needed, the range needed, the distance it might work, and the impact of that. So we looked at two focuses really, I suppose. What conjugates would we stick onto monoclonal antibodies if they wanted to work on a tumor mass, like in this diagram? Or what radioisotopes would you consider for imaging of tumors? So we went through the different types of radioisotopes. We looked at the different ranges, the different distances that they cover. And we talked about how we can even look at, I suppose, some new ways of trying to minimize the radioactivity doses, even like in a two-stage process, and how people are trying to find new ways of this, like even click chemistry, things clicking together so that the radioactivity will only be at the right place. We talked about pre-targeting, trying to get around a problem in certain situations, and we went on imaging, the idea that we might have used a gamma source, gamma ray emission. It can penetrate the body, so it can get out to the camera, but it has a short half-life of 6 hours. And we talked a little bit about how for example, one that's looked at an awful lot is this CEA scan. It's a FAB fragment. So remember the antigen binding regions of a specific murine monoclonal antibody against this CEA. And CEA is expressed by an awful lot of gastrointestinal tumors. So when we reflect at this stage, the first section I wanted to cover with you was really about biopharma versus pharma. And just a summary, remind you of some of the things you've done in other modules and how you've been learning about this throughout the last couple of years already, bits and pieces, but try and tie it all together. And then we focused in on a protein family called cytokines, how they work through receptors, intracellular signaling, and ultimately cause biological effects in the cells that they've bound to. So once we had those concepts, we were able to focus on a family known as the interferons. And those interferons, we were going to try and go through the idea of how they function, what they work, what their role would be. And I gave you a handout that I wanted you to go through and isolate or identify the biological effects of those. We moved on to a new handout then on hematopoietic growth factors, focusing on erythropoietin. And this was a really good one to go through because we talked about recombinant DNA, we talked about glycosylation, and we brought back the idea of when we need things, turning on things as we need them, and then how they go to do their job, and ultimately how half-lives of proteins are dictated, and how we can, I suppose, revisit even the biosimilar approach with designing new things. And then finally, we went through the whole idea of antibodies. You learned a lot about these in immunology, but now we've learned a little bit about how they can be produced as monoclonal versus polyclonal, and that difference in responses. We learned a little bit about conjugates that we can stick on to the antibodies, and how the antibodies will bring whatever we stick onto it to the target. And we went through those different types of radioisotopes that could be used in different circumstances. So I hope that gave you an overview to some of the things that we've talked about in the first few weeks. Okay, hope all is well.